to start with some fairly simple but important observations. And the observations are that language is an ability that is uniquely human. It has a highly complex hierarchical structure, and humans easily understand that structure on the fly. Meaning that speech sounds combine into parts of words, which create words themselves, words combine into phrases, phrases combine into sentences, and we understand language without giving this a second thought. This is an unconscious process. And the hierarchical structure present in language arises due to the dependencies, or in other words, statistical correspondences, between linguistic units at different levels of language organization. So, for example, think about the relationship between the form of the verb and the subject in English present tense. So compare sentences like they paint to she paints. The ending of the verb reflects the subject. And a more familiar term for this is just grammatical rules. All human languages, as anyone who's learned a second language knows, have highly complex grammar. And no other animal has a communicative system or communicative abilities rivaling humans in their complexity. And that makes it important to understand the neurobiological processes underlying our ability to comprehend grammar on the fly. And recent work suggests that rhythmic neural activity might help answer this question, as changes in oscillatory behavior seem to track structure at different levels of language. So, for example, in their 2015 paper, Ding and colleagues find that cortical activity of different timescales concurrently track the time course of abstract linguistic structures at different hierarchical levels, such as words, phrases, and sentences, meaning the following. In the experiment, Mandarin participants were presented with 4-word sentences, which consisted of two word phrases, and their brain responses to these sentences were recorded with MEG. All words were monosyllabic and presented isochronously at 4 hertz, and the experimenters found that at the acoustic level presented in B here, the stimuli contain a spectral peak at 4 hertz, but no peaks at 2 or 1 hertz. And this was different compared to the MEG-derived cortical response, which you can see in C. It did have peaks at all 3 frequencies, corresponding to the frequencies of word, phrase, and sentence presentation. And later, they conducted further experiments testing whether these peaks arise because of participants' knowledge of the grammar of the language. And they do see that that is indeed the case. So, for example, in A, you can see that when participants are presented with just random words, which are not grammatically connected, you only see the peak at 4 hertz. When in B, you can see that when participants are presented with just phrases, which are not connected into sentences, you see a peak at 2 hertz, but not at 1 hertz. And if you present the same materials, but to English listeners, which you can see in D, then you only get an increase at 4 hertz. So obviously the knowledge or the structure changes in some way grammatically, and participants brain respond to. And another study coming from the same group showed that this effect can emerge quite rapidly. In an implicit artificial grammar learning experiments, Getz and colleagues presented to the participants sequences of meaningless syllables for 40 minutes. Each syllable uniquely belonged to one of 6 categories, and for half of the participants, the artificial grammar, the sentences in the artificial grammar was structured in such a way that they contain strong statistical correspondences between some categories, which you can see in the red here. So, for example, words from category A were always followed by category B words, C was always followed by D, and E was always followed by F. And this type of structure is illustrated in the red graph here on the left. The artificial grammar presented to the other half of the participants did not contain such strong distributional cues, and word categories could follow each other relatively flexibly. And you can think of the red or phrase structure condition as being relatively analogous, even though being significantly more simple, to how phrases are structured in languages. So, for example, if you hear an article like the or an, you're likely to expect an adjective or a noun to follow it. And Ding et al. Found that this 14-minute implicit exposure session was enough to elicit an effect similar to the one found in real languages. Both artificial grammars elicited an equally strong spectral peak at the word presentation rate, but the phrase structure condition resulted in much more significant peak at the 2-word or phrase presentation rate. And what we decided is to leverage this methodology to answer 2 questions important in understanding how humans process grammar. First, how domain-specific is neural rhythmic activity tracking of hierarchical structure? And second, how unique to humans is neural rhythmic activity tracking of hierarchical structure? And the answers to both of these questions could help progress the debate on whether humans have some special cognitive ability that helps us understand language that we don't share with any other animals, as some linguists, cognitive scientists, and neuroscientists theorize, or whether the process of language comprehension is ultimately built using a more general cognitive toolbox that we share with other animals. And we address these research questions using the following approach. We follow the methodology of the artificial grammar learning experiment that I described earlier by Getz and colleagues. We have two types of grammars in terms of their structure. Both grammars have 6 categories of stimuli, but they differ in terms of transitional probabilities between them. Whereas in the unstructured artificial grammar, which you can see on the left, the categories follow each other relatively freely and don't create any distributional cues. In local dependency artificial grammar conditions, the categories are organized into 3 pairs, such that the categories always co-occur next to each other in a pair, one always preceding the other. This creates a distributional cue due to the 100% transition probability between them. For example, as you can see in this specific case on the right, if you hear a stimulus from the category A1, it will always be followed by a stimulus from A2. And in our experiment, we ensure that in the unstructured artificial grammar, there is a uniform probability of transition between all categories across the whole exposure session. Additionally, we manipulate whether the stimuli presented to the participants are meaningless words or drum sounds, allowing us to see whether the effect can be found for non-linguistic stimuli as well. And furthermore, we record EEG from human participants and LFP and EEG from rat participants. And you'll hear more about this setup in the next talk. We do this in order to compare the results across species. Each exposure session is composed out of 27 blocks of sentences, each block containing 33 4, 6, or 8 stimulus sentences separated by a quarter of a second pause. The stimuli are presented isochronously with a 4 Hz presentation rate, and each exposure session lasts half an hour, during which participants listen to the stimuli and are not given any additional tasks. So currently we have data from 5 rat subjects and 7 human subjects and are still in the process of collecting data. Additionally, we see a lot of variability of data. Because of that, we won't draw any strong conclusions in this talk. However, some patterns we observed do seem to be quite suggestive. We analyze the data using linear models and, if possible, using mixed effects models with random effects. In this presentation also, I will particularly focus on results from rats. So if we plot the results from all rats, we see the following. In this and next graphs, colors denote stimulus type and x-axis denotes types of dependency. Y-axis here denotes the average power from all of the 16 LFP channels reported from the rats. And I will not be presenting results from the EEG recordings in this talk. And here we can see that at 2 Hz, there is relatively more power for the local dependency artificial grammar compared to the unstructured artificial grammar. However, surprisingly, we see the reverse pattern for the drops. There is more power for the unstructured artificial grammar compared to local dependency, thus creating the sort of interaction effect. And furthermore, we also see the same effect at 4 Hz, which was not that expected. However, as I mentioned, there is a lot of variability between participants, and the data from some rats doesn't look like data from other rats. For example, for this specific participant, we see the pattern is quite reflexive and quite similar to the overall pattern. However, it is not reflective of the pattern that we see in other rats. So, for example, in PR18, we see quite a different pattern. We see a significant effect of dependency type. So local dependency conditions elicit more power in the recordings and more so at 2 Hz compared to 4 Hz. And most of the comparisons between local and unstructured conditions matched on other factors also result in statistically significant results. And most rats show patterns actually more similar to PR18 than PR23. And so if we remove PR23 from the analyses, we see quite a different pattern overall. Now we see that overall at 2 Hz, we see more power in local dependency conditions, both for drum and for speech stimuli. And we also see an increase in power at 4 Hz in the local conditions. However, it is much less strong. Overall, while the results from rats are preliminary, and we definitely can't draw any strong conclusions, they are suggestive of the possibility that rhythmic neural activity could track hierarchical structure, not just in humans, but in other animals as well. And for humans, there's a lot more variability from participant to participant. And overall, we don't see the same pattern as in rats. So I won't present the data from all the participants. We don't really see an increase in power in the local dependency condition at 2 Hz compared to unstructured conditions. However, in 2 of the participants, we see a pattern that is reminiscent of the pattern that we see in PR23 in rats. Specifically, this interaction where at 2 Hz frequency, we see more power for drums for local dependency, but more power for unstructured dependency for speech. We're currently investigating the reasons for this increase. There's a lot of different possibilities that might be quite pragmatic. It might be due to the presentation order and participants fatiguing, or potentially this will, it also could be a more interesting possibility is that some people show a different pattern of tracking structure through rhythmic activity and have a decrease in activation. While this would be surprising, such patterns of results are not unheard of, and we do see similar patterns of results in language ERP studies, where, for example, in response to the same violations, some people show positivity, where other people show negativity.
You're at 10 minutes, Arturs.
So yeah, as just a conclusion, the main finding that I want to focus on is that we see some evidence for tracking a hierarchical structure in language and other types of stimuli in non-human animals' rhythmical brain activity. But we need to conduct further work to investigate this. And just to conclude, I would really like to thank all the incredibly smart and helpful people who have worked on the project as well, and without whom this presentation would not be possible. I'd like to thank Pauli Institute for Brain and Mind for funding the project, and thank you for listening.
Great, thank you. Really interesting. We have a question from Terry. Were the human subjects consciously aware of the local dependencies?
So we didn't give them any instructions beforehand, and afterwards I spoke to a couple of them. Some of them noticed it. One person didn't, but it's pretty hard to kind of like miss if you actually like listen to it. Yeah. But they weren't given any explicit instructions to look for them.
And a question from Eric Hallgren. Did you look at the event-related potentials between the conditions?
No, that is one of the next steps that we're planning to take once we collect more data, but we haven't done that so far.
And then Zoe Cheng, is comprehension level measured in this study in both humans and rats?
No, so we decided not to do this just for the ease of comparison. We can expand that in the future experiments, but right now we're not doing that.
We have a couple more minutes if there are more questions anybody wants to add into the chat. Am I understanding correctly that the next talk from Taryn is going to follow on this or be related in some way, Arturs?
Well, it's not going to be related, but it also uses the same EEG LFP.